Hello and welcome dear viewers to the video on initial and shelf life testing for pharmaceuticals. So shelf life testing means it involves the stability testing. Initial testing involves the routine test onto the formulation to release the batch in the market and also to release the batch for stability studies. If the formulation batches are exhibit batches or validation batches, and for the formulation batches for which or the product for which the stability is to be done, stability studies are to be done for regulatory filing. For those, initial testing is done for performing the stability studies or to load or charge the product batches onto stability studies. Initial testing is also must for selecting the formulation batch for bioequivalence studies, also for bioavailability studies. So this video will be a short information on initial and stability testing for pharmaceuticals, and mainly we will consider the tablet and capsule formulations. So in general it will be initial and stability testing. Now the drug product testing at initial and during the stability intervals and after the storage period and shelf life period is required. So whenever you are taking the batches for regulatory filing purpose or you are demonstrating the process capacity for manufacturing, also whenever you are doing the scale up and post approval changes for the formulations which are already approved, or whenever you are going to do the site transfers, that time the stability studies are required. So these studies are performed onto the stability testing intervals on accelerated, intermediate and the long term conditions. Initial testing is done for generation of the certificate of analysis, release the batch in the market if it is a commercial batch and it is approved. Then COA is required for batch release. Also the initial testing and initial COA is required to load the batches onto stability studies for regulatory filing. And in this video we will consider the tablet and capsules as a formulation, that is oral solid formulation for initial and the stability testing. So the test will be there and the specifications will be there. So here one thing is to consider is that what are the tests to be performed on initial and what are the tests to be performed on the stability studies. On initial, extensive studies are performed, and after meeting the initial specifications, then only the batches are loaded onto the stability at different conditions as per the product requirement and the regulatory body requirement. So at initial, the description is tested, the physical appearance of the formulation is checked, whether it may be tablet or capsule. Then average weight. Then identification is done by HPLC and IR. So identification test involves the testing which involves the two principles like HPLC and the IR. So these are the two different principles. Then uniformity of weight and uniformity of dosage units, that is also known as content uniformity. Then after the content uniformity, the dissolution testing is performed. Organic impurities or related substances testing is performed. You can remember it as impurity testing or impurity profiling at initial stage. Then residual solvents are required to be tested. If the formulation involves the use of residual solvents, that time the batch release testing involves the test for residual solvents. For example, if you are using isopropyl alcohol for batch manufacturing, so you have to test for the isopropyl alcohol. If at all the formulation, composition and manufacturing process does not involve the use of residual solvent, that time the compliance to the ICH and USP is required, and that will be as per the option 1. Then assay testing is done. What is the content of the tablet or capsule as compared to the label claim? So for US, the limits are 90 to 110% of the label claim, and for Europe it is 95 to 105% of the label claim. So that is the difference. Disintegration testing is performed. Then microbial testing. So for oral solids, the microbial testing is done, and it involves the testing for yeast and molds and specific organisms like E. coli. So this is for the initial testing and batch release. Now additional testing is also performed at the initial stage. So the additional testings are performed to understand, is there any impact of the manufacturing process, and also these additional tests will help you to understand the product behavior in the stability testing or during the stability and shelf life. So additional testing are water content or LOD, any process impurity if it is required to be monitored or if it is required to be tested. So if you know that the process impurity is there in the API, and if you want to monitor it to understand whether it is process plus degradant or it is only process in the formulation also. Any chiral impurity or enantiomeric impurity, that impurity is required to be tested through the additional test. And another is hardness testing for the tablets. Sometimes the formulation of tablet may fail in the stability studies in dissolution. So that time you have the data of initial and the stability testing for hardness test. If the dissolution is failing because of the increase in hardness, you can correlate these things and you can move on. If the water content is increasing, then you can say that water content is increasing. That means you have to change or monitor water content, and also you can change the packaging system. Then stability testing. So in the initial testing, extensive testing is performed, while in the stability the tests are less. So description is there, assay, dissolution, organic impurities, disintegration testing, and the microbial testing is done. And why these tests are performed into the stability studies? Because on storage period or on standing period or not standing period, you can say it as shelf life period, or aging of the formulation. So description may get changed. If it absorb water, that time description may get changed. Sometimes the color variations are there in the capsule formulations compared to initial. Sometimes the capsule shells may become hard or may break because of loss of moisture or gain of moisture. Sometimes microbial growth may be observed. That's why these testings are done for stability testing. Assay may increase or decrease during shelf life period. dissolution may get decreased. There may be a chance of increase in the organic impurities. There may be a increase in disintegration testing, and there are chances of microbial contamination and the microbial growth. So these tests are done on the stability period or onto the formulation in stability. Then additional stability testing and the tests are like water content, any process impurity if required to be tested, any chiral impurity or enantiomeric impurity, and the hardness. So these tests are the same as the additional test for initial analysis. Then only you can understand the effect of different storage conditions and the packaging systems during the shelf life. Now we will compare here which are the tested included in both initial and stability test, and which are the tests which are only for the initial. So description is for both initial and stability. Average weight identification, uniformity of weight, and CU, that is content uniformity, is only performed onto the initial. And the reason for performing it on initial only and not in stability might be that these parameters may not get changed much due to storage or stability. While dissolution is tested, organic impurities are tested, that is related substances are tested. Residual solvents are not tested in the stability because once you control the residual solvents, these will not increase on storage. Assay is of course required to be tested. Disintegration testing is done and microbial testing is done. So some of the tests are done only on the initial and some tests are done on both initial and stability period. So this is regarding the initial and stability testing of the oral solid formulations. The main motto of preparing this video is that many of the professionals don't understand what are the tests for initial and what are the tests for stability. So assay, related substances, dissolution, and water content for some of the products is tested on stability period. Sometimes some of the applicants test disintegration testing, test the formulation for disintegration in stability, and for some of the formulations like tablets, hardness is required to be tested. So DT and hardness may be included in the release specification, or you can do the additional testing for the hardness test. So additional testing what is additional testing? Additional test in the initial and the stability period are those tests which you are not going to file or you are not going to include into the specifications, but you want to generate the data or for these tests, like for checking the disintegration and hardness. The reason is to correlate with these tests with the dissolution, and then you can have the good understanding of the product behavior. Also these tests are performed to select the best packaging configuration and the packaging materials. If your product is failing in PVC blister and if it is passing in alu blister, then with the additional test along with the main stability and release test, you can select the packaging. If you come to know that in some of the packaging hardness is getting increased and that's why, or because of the moisture gain hardness is changing and disintegration time is getting lengthy, and that is because of only of the packaging system, then easily you can identify the best packaging system and you can go for filing the product. So I hope I have covered uh, the information on initial and stability testing, and I hope you might have got good knowledge out of this video. So thank you for watching the video, and I request you to share these videos from Pharma Learning in their channel to your friends and colleagues. Thank you.